All right, let's go through our cell reproduction or mitosis video. Um, all of us. Okay. You need to be able to basically describe the essential properties of cell division in prokaryotes. Remember, a prokaryote is a very simplistic, ancient sort of cell. A lot of bacteria prokaryotes, right? They're defined by the fact that they don't have a true nucleus to them. The key thing that we need to be able to do is describe cell division in eukaryotes. Those are animal and plant cells that are defined by the presence of a true nucleus. You should be able to explain the key differences. You need to be able to talk about chromosomes. Chromosomes are the way in which DNA is packaged in the cell. You need to be able to describe the key steps in the cell cycle. You need to be able to identify and describe the main steps and processes in mitosis. That's PMAT. And then we'll briefly talk about cancer and how cancer is the cell cycle run amok or afoul. Cell division divides a cell to form two new cells. These new cells that are made are called daughter cells. There are three main types of cell division. Prokaryotes, the bacteria, right, go through a process of binary fission. This allows for a kind of exponential growth curve for forming new bacteria. We did that demonstration about how that works with our bingo chips. It's essentially a process of cloning itself. Eukaryotes go through two different kinds of cell division. The cell division that we're interested in here is called mitosis, and this is a form of copying the cell. In a way, it's a kind of cloning of itself too. This allows for you to grow and develop, right? Becoming a child to an adult, right? You're growing new cells and growing taller and bigger. Also, if you become injured, you can replace or repair damaged cells as well. The next process is meiosis, and we'll talk about this later. This is how sex cells are formed, sperm in men and eggs in women. The sex cells are called the gametes. So we briefly talked about why do cells need to divide. Because they continue to grow in size, and if the cell continues to grow in size, then the DNA will no longer be able to control the functions of that cell. Also, the cell membrane, as we talked about before, allows materials to pass in and out. If it becomes too large, then it will impede this process. It will go too slowly. Needed materials for metabolism won't be able to enter the cell, and waste products of metabolism won't be able to exit the cell. So prokaryotic division is known as binary fission. It occurs in three steps. The first step, DNA is replicated. Replicate means to copy. If you don't know what this word replicate or duplicate mean, you need to take the initiative and look it up for yourself. You need to be proactive and look it up. It means that the DNA is copied and we have two identical chromosomes. Once you get into AP biology, we'll spend a lot of time talking about DNA replication. But for now, all you need to know is that the DNA is copied. The next step is that the chromosomes segregate. The chromosome in bacteria is usually a circular one known as a plasmid. They separate to different poles of the cell, which means opposite ends, just like the Earth has a north and a south pole. The chromosomes migrate to either end. And then we have cytokinesis. Cytokinesis. That's when the cytoplasm divides, right? The cytoplasm is anything that doesn't involve the nucleus of the cell. And we get two new cells that are formed. And each new cell is exactly the same as the original one. They are genetically identical. They have the same DNA. Okay, so this is a great picture of the cell cycle. As we can see, most of the time cells are either in G1, which stands for growth, cell growth, or the S phase when DNA replication occurs. And then there's a secondary growth phase as well, G2 for growth as well. The G1, S phase, and G2 phase all make up what's called interphase. Okay? These are three different things that happen during interphase. So the first part of interphase, we have a new cell, it's growing larger, it's developing, and then it reaches a certain size, and it says, well, in order to function properly, I need to divide so I can reduce my size. So that goes into the S phase or synthesis phase when DNA is replicated. That takes a while. And then we've got the G2 phase when other organelles in the cell are replicated as well. And we get ready for the M phase. M stands for mitosis, okay, which is a part of cell division. After mitosis occurs, we have cytokinesis, right, which is the separating of the cytoplasm and the pinching off of the membrane to form two daughter cells. Okay. Mitosis has 4 steps: prophase, metaphase, anaphase, and telophase. You need to be able to recognize which stage of mitosis a cell is in. Okay, so let's go through the phases briefly. So interphase, which is the longest period that a cell goes through, right? It's the period where the cell grows and it replicates its DNA. Once it reaches a certain size, it knows that it needs to replicate. As I've mentioned, interphase happens in 3 phases. G1 phase, again, G stands for growth, where the cell increases in size. S phase, S stands for synthesis. Synthesis is a fancy word to say make. What's it making? Well, it's making more DNA. It's replicating its DNA. DNA is found within structures called chromosomes. And when we have a replicated chromosome together and they're joined together here, these are called sister chromatids. In the middle where the two, so you've got the original chromosome and then you've got the replicated one. So once the DNA is found within the chromosome. Okay? Once it replicates, it later becomes condensed in a form which you can see it as the chromosome. Chromosomes are attached in the middle by the centromere and a special kind of protein structure on the centromere. It's called the kinetochore, which attaches later to the spindle fibers. In the growth 2 phase, in the G2 phase, the other organelles double and we need to make new cytoplasm and other structures that are needed for mitosis form. Okay, so going back, what is a chromosome? A chromosome is a way in which the DNA is packaged, right? The DNA is all the genetic information, all the instructions, or if you will, the code to make you. Each chromosome has a matching pair. So in humans, there are 23 pairs of chromosomes. So that means 46 altogether. Each chromosome in a pair is inherited from each parent. So one of them comes from your mother and one of them comes from your father. These pairs, the one that comes from the mother and one comes from the father, are called a homologous pair. Okay? During interphase, each chromosome copies itself, and then once it's become copied, they're now referred to as sister chromatids. sister chromatids. Okay. So eukaryotic cell division, right? This is not binary fission. This is what we go through with mitosis and cytokinesis. Okay. Regularly, right, the DNA is found within the nucleus of a cell in eukaryotes, right? And prokaryotes, right, they don't have a nucleus. The cell cycle is a continuous process. It's a circle. It's a cycle. It starts over and over and over again. Okay? The process, cells grow, G1. DNA is replicated, S1. Organelles duplicate, which means replicate. They make a copy, a second one. That's G2. And then we've got the cell division, right, which is mitosis and cytokinesis. And then once that happens, it starts all over again. The cells growing, G1, S, G2. Mitosis and cytokinesis, then start all over again with G1. It's a cycle, it continues, it repeats. Okay? Mitosis has 4 steps, and you need to remember these. Okay? The easiest way to remember it is by remembering the acronym PMAT. P for prophase, M for metaphase, A for anaphase, T for telophase. PMAT. PMAT. Okay? And then after mitosis comes cytokinesis, where the cytoplasm divides, pinches off, and you have 2 new cells. But the most important part of the whole thing is duplicating the DNA and getting the DNA, the exact copies of the replicated DNA, into each new cell, each new daughter cell. Okay, mitosis is a form of nuclear division. Mitosis is always followed by cytokinesis, cell division, and there are exact steps that it follows every time, right? This is the great thing about science is that it's observable and it's repeatable. It continues on and on. Right, we want to, because the whole point of mitosis is we want to ensure that each cell is an exact copy. It has the exact same instructions or genetic information as the parent cell did. If every cell has different instructions about how to behave inside of it, your body would be in chaos. Nothing would be able to get done. Remember, when we were talking about one of the key characteristics of life, one of the key characteristics of life was homeostasis. And that's the ability of an organism to maintain a consistent internal environment, right? One of the examples we used was with body temperature. Body temperature has to stay at the same temperature. It can't go up, can't go down. Okay. So here is an overview summary of mitosis and the 4 stages. The PMAT, prophase, metaphase, anaphase, telophase. Okay, and you can see the distinguishing characteristics of each. And spend a lot of time practicing so that you can see which one it's in. Okay, you need to be able to identify which stage that it's in. You need to be able to draw them too. So on a test, you might have to identify from the picture or to be able to draw each stage or talk about what's happening in each stage. So the first stage of mitosis is prophase. Okay? Remember, most of the time, the DNA in a cell during interphase, right, is not visible, meaning light passes right through it. You can't see it. It's small, tightly wound structures called chromatin. It's the way it usually is. Okay. But during prophase, the interesting thing is the DNA and the structures become condensed, meaning that light bounces off of them. We can now see the actual chromosomes, right? The chromosomes have been replicated at this part, right? They are now as sister chromatids. They're these X structures. These are the sister chromatids. One of them was one of the original ones from the cell, and the other one is a copy that it made during the S phase of interphase. Okay? There are these structures here called centrioles. Okay? Centrioles migrate to the poles of the cell. So they move either to, to the right and the left side. Okay? These are the structures that have these spindles attached to them, or spindle fibers. And the spindle fibers attach to the centromere, the central area of the joined pair of sister chromatids. Okay, the spindle fiber acts like a rope to pull it apart. Okay, the centriole is kind of like a gear. It turns, it pulls the different chromatids apart. Another distinguishing feature of prophase is that the nuclear membrane disappears, and we can see it starting to dissolve here. And you can begin to see the spindle fibers. So that's prophase. Metaphase, the distinctive feature of metaphase is that all the chromosomes, they line up in the center of the cell. Okay? This part where they line up, it's called the metaphase plate. Okay? We've got the centrioles in either pole, and they've got spindle fibers that are attached to that centromere in the middle. Remember, there's that special protein that makes up the centromere called the kinetochore, and that's where it actually attaches. Okay, anaphase. We can see the sister chromatids are being pulled apart, and the chromatids are now going to migrate to either pole, right? This spindle fiber is like a rope, and the centrioles are pulling them. They're pulling them apart to either pole. Okay, telophase. The sister chromatids are in either pole right now. We can see that the nucleus is beginning to reform in each cell, and we can see that the cytoplasm and the membrane are starting to kind of pinch off in the middle. This is known as a cleavage furrow, this kind of cleavage furrow in the middle. We can almost see two cells, where it's got this kind of dumbbell shape. And these kind of spindle fibers begin to disappear as well. Okay, cytokinesis is the last step. Cyto refers to the cytoplasm, and kinesis is kind of the word for kinetic, which has to deal with movement. Okay, this happens at the end of mitosis. Everything divides, and now we've got two, and these are referred to as two daughter cells. Okay, the organelles, right, that were replicated in the G2 phase of interphase, they've now nicely divided as well. Remember that the cytoplasm refers to both the cytosol and everything in it. So it's the liquid part and everything in it as well. It's not just the liquid part. The liquid part is known as cytosol. Everything except for the nucleus is considered to be the cytoplasm. Okay? So this is a cycle, part of the whole cell cycle. So after this is complete, right, everything goes back and returns to interphase again. And our animation didn't really work. So you want to be able to quiz yourself, because you might see a picture like this and you need to know what phase it's in. Okay? So the first one here at the top, this would be interphase, right? Except, so we've got this kind of mashed up DNA in the center of the nucleus there. The DNA at this point is known as chromatin. Chromatin. Okay? This one here, the DNA has obviously replicated, right? We can see the sister chromatids. We can also see that the nuclear membrane is beginning to dissolve. We can see the spindle fibers and the centrioles are beginning to migrate to each pole. This is prophase. Prophase. Okay. This is a beautiful example of metaphase. All the sister chromatids are lined up in the center, known as the metaphase plate. Okay. This is anaphase. It kind of looks like a watermelon in a way. Okay. You've got the sister chromatids are being pulled apart. They're being pulled to opposite poles of each cell by the centrioles. Okay. Telophase. Look, we've got the DNA, we've got the chromosomes on either side now. Sister chromatids have gone to either side. We've got a nuclear membrane that's reforming in each one. We've got the cytoplasm and cell membrane pinching off, forming this cleavage furrow here. This is telophase, right? Telophase is finished, right? Cytokinesis happens. It finally pinches off and it goes back to interphase. Interphase, prophase, metaphase, anaphase, telophase, on and on. Okay? So what's this one? Oh, they're being pulled apart. So this is anaphase. Anaphase. What's X? X is a spindle fiber. What's Y? Y is a centriole. What phase is it? Well, we got the chromosomes on either side starting to kind of pinch off in the middle. This would be telophase. What phase is this? So we've got centrioles, we've got the chromosomes are visible in the pair of sister chromatids, we've got a nuclear membrane that's beginning to break down. This is prophase. Prophase. Okay, Yeeler asked a really good question the other day about how long does the cell cycle take, and I'm not sure if I gave a really good answer to this. So it can last for a lot of cells about 24 hours, one day. Okay. For different cells, it can last longer. So for example, like liver cells do take a lot longer to be completed. But if you are like a mosquito larva, this process can occur within 8 hours, much less time. So if we look at it, the cell spends most of its time in G1, growing, developing. The second most time it spends is in terms of replicating its DNA in the S phase. Mitosis is the shortest period. Okay, so how does it know which stage to come in? Well, through most things that happen in cells, we talked about this in our first unit, proteins decide what goes on through kind of chemical interactions. We don't need to know exactly how it occurs, but there are proteins that kind of regulate the whole cycle, and they are called cyclins. And they check where the cell is at certain points of the cycle. So there is 3 different checkpoints where it decides if it's going to divide. So there's the G1 checkpoint, so it decides whether or not it will divide, right? The cell needs to be at a certain size, right? And it says, oh, we've reached that size. The cyclin can determine that. Let's move on to S phase, the S portion of interphase, and start replicating the DNA. Okay, once the DNA has been properly replicated, another cyclin comes by and says, okay, this stage is complete, it's done properly, we can move on to mitosis. And then there's another one. This is the mitotic plate, so that's the metaphase plate there. Okay. So it shows you the 3 different checkpoints. Okay, so G1, right, is it big enough? If it's okay, then move into S phase. If not, then keep growing and dividing. Okay? S phase, right? It checks, has the DNA been replicated? And not only that, this is a thing for AP biology, it's not only it needs to be replicated, but it needs to be replicated correctly, which means exactly. It must minimize errors. If errors creep into the DNA, then you have proteins that can't form correctly and cells that don't work correctly. and you have a lot of problems. So there are proteins that check DNA as well, but this cyclin just checks, has the DNA been replicated and has it been replicated correctly? If so, boom, we move on to the next stage. G2, we're going to replicate those organelles, and then we're going to move into mitosis, right? And the big major part of mitosis is, right, those chromosomes need to line up at the metaphase plate, and then through anaphase they can start separating. So if they're lined up correctly at the plate, then the cyclin will say, yep, check, boom, move into telophase, right, and then cytokinesis, and then start again. So these are 3 different checkpoints where it decides if it needs to move through. Okay, so what happens if something goes wrong with cell division? Well, sometimes cell division can become uncontrolled. It doesn't go through this normal process. Those cyclins then have lost the ability to regulate the process, and they've gone uncontrolled. Okay. What happens with normal cell division is it continues on and on and on, but then maybe there's some sort of cell damage, or the DNA hasn't replicated correctly. There's something that went wrong with the whole process during the cell cycle. So what cyclins can do is just say, okay, well, this cell is not good. If it continues, we're going to have huge problems, because there's problems with the instructions, maybe the chromosomes are in the wrong place. What it does is that the cell goes through a process of programmed cell death or cell suicide. This programmed cell death is what's known as apoptosis. Apoptosis means cell death. But what can happen is that sometimes this doesn't work, and we've got damaged or sick cells that continue to replicate, and they don't shut down because the cyclins aren't stopping them. So we can imagine what could happen. So you get these errors, and they continue to grow, and maybe you can get more errors and more errors and more errors. These are cancer cells. Okay, they're the cells that aren't functioning properly. And what they do is they can stick together and form these masses, and masses of tumors, are called tumors. The good thing is, if you catch it quickly enough, surgery, you can remove a tumor. However, most cells have proteins that make them kind of sticky or adhesive, and they stick together. But if damage continues to go on, then they can become less sticky, and then pieces can float off and go to other parts of the body. And if they start floating off, going to other parts of the body, then you get tumors and growth everywhere, and you're in serious trouble. That's called metastasis. Okay, so what should you have learned from this? That there's a cell cycle that governs the life of a cell. Okay, it's a repeating series of events. This is found in eukaryotic cells. Prokaryotic cells carry out some form of binary fission. They just keep dividing by themselves. They kind of are a bit like cancer cells in that way. When eukaryotic cells want to divide, the DNA and proteins together, they kind of coil up and they form chromosomes. That's the visible DNA as a chromosome. DNA copies itself during S phase, and we've got 2 identical chromosomes, and these are called sister chromatids, and they are attached together in a kind of X shape in the middle called the centromere. The next point, I'm not why I included it, but mitosis makes diploid cells, meaning they've got 2 copies of each. They're not copies, but there are 2 homologous chromosomes. There's a chromosome pair. One comes from the mother, one comes from the father. When we look at meiosis, we'll see that the process is similar in many ways, but it produces haploid cells. They have half the amount of genetic information, which is important for producing children. The cell cycle goes through growth and synthesis and a second growth phase, and then mitosis and cytokinesis. Mitosis divides the nucleus into 4 distinct phases, known as prophase, metaphase, anaphase, telophase. Memorize PMAT. And then we have cytokinesis, which divides the cytoplasm. Okay? There are feedback mechanisms that control and check the stages of cell division through proteins called cyclins, and they check at 3 distinct spots in the cell cycle. And then cancer is the result of the unregulated cell cycle. Okay, and you get cells that have become damaged but continue to replicate. There's no apoptosis. There's no programmed cell death. Okay? There's a lot here, and it does take quite a bit of study, but after a while, you begin to see the different stages. We're going to do lots of practice in class, but you still need to be practicing this during your evening study hall. Good luck, biologists.